Great. Well, thank you so much for the opportunity to share this work. I'm really excited to talk about it. So today I will be talking about the role of phase separation in SARS CoV-2 packaging, and specifically really trying to address the question of how does the nucleocapsid protein specifically essentially find, identify the genome in a sea of RNA, interact with the genome, and then package it. And my interest in this problem comes from a longstanding and deep interest in understanding how the cytoplasm is organized. And this is really a frontier in cell biology. The cytosol is a mysterious environment still in terms of how it's spatially organized, and how within this incredibly dense and dynamic milieu do you have organization emerge. And so I'm going to take a few minutes to share with you just the principles that are emerging for cytoplasm organization with regard to a mechanism of organization called phase separation. And I'm going to talk about the mechanics of this a little bit before I actually get into how this relates to SARS biology. And so how can order emerge in the cytoplasm? It's now increasingly appreciated that molecules can self-assemble and essentially condense into compartments that have liquid-like or solid-like properties, just like water can exist within a soluble liquid or solid-like state. And this happens both in the cytoplasm, but also, of course, in the nucleoplasm. And that this is a new way of thinking about ways of organizing biological molecules in cells. And it's now appreciated that biological condensates are a nearly universal feature of cells that serve many purposes, from long-range transport in neurons to enabling cells to react to stress and the formation of stress granules, also relevant probably to SARS replication, to the patterning of embryos and generating asymmetries within cells, to name just a few examples. However, there's also increasing evidence of phase separation being relevant for replication in virology. And I'm just showing two of the most recent examples of this from the literature in 2020 of the N-protein from measles undergoing a liquid-liquid phase separation. That is, it's able to demix from the surrounding milieu and form compartments that have liquid-like properties. Similarly, the GAG protein from retroviruses, here this is looking at HIV, is able to self-assemble into these liquid-like compartments. And here you can see this being reconstituted in vitro and also in cell expression studies. So what do phase-separated condensates actually do? This is an area of intensive study. The basic mechanisms are thought this is a way to bring factors together in order to interact at the right time and the right place, that they serve as vessels for chemical reactions, that they have the capacity to enhance enzymatic rates, and importantly can act as stress sensors of temperature and pH, because they themselves are often physiochemically driven. There's a great potential for serving as stress sensors. So I want to give you a bit of just 101 on condensate formation before I get into how this relates to the SARS. So how do condensates form? In many cases, they involve a nucleic acid binding protein. So this is similar to nucleocapsid. So in many cases, it's an RNA binding protein that also has a highly disordered or low complexity sequence. And this means that the protein can take on many, many different conformations or shapes rather than a stereotypical secondary or tertiary structure. Many condensates also contain nucleic acid, and in many cases, this is an RNA molecule. And so the capacity of an RNA binding protein to interact with an RNA molecule is absolutely critical. And that's because this allows for a multivalent interaction. So essentially, there's multiple sticky spots within these proteins that allow protein-protein interaction through disordered sequences interacting, but also for the RNA binding domain to interact with specific and often multiple sites on an RNA molecule. And so when there's a sufficient number of these molecules with this capacity to interact, This drives the condensation process, which ultimately leads to the emergence of mesoscale droplets that we can detect and see under the microscope. And depending on the strength or the affinity of these interactions, the spacing of the sticky spots on the RNA or the protein, this will inform the material properties of the droplets. And it's thought that the material properties is really relevant for their function. And so material properties that we can measure and think about are the viscosity, the ability of droplets to fuse. This will influence things such as the size of the droplets or the sphericity of the droplets. And so essentially nanoscale features of protein and RNA interactions ultimately lead to condensates that have particular physical states. And one of the powerful aspects of studying phase separation or condensates in biology is that this can be reconstituted in a very simplified system, often just by mixing together the proteins and the RNAs together. You can have a spontaneous demixing occur. And in my group, we're really focusing on the RNA or the nucleic acid component of condensates. And there's a lot of really interesting features of RNA that they are, we think, contributing, that RNA molecules are contributing to the features of condensates. But I should say these are very, very understudied. So RNA, of course, has a uniform negative charge. There's the potential to encode multiple binding sites for a given protein, thereby providing multivalency. They are variable and also incredibly large in size relative to proteins. And obviously this is highly relevant with the gigantic 30 kb genome that we see with SARS CoV-2. RNA can also assemble in a variety of dynamic structures, the tertiary and also quaternary level, that are sequence encoded and can be remodeled by enzymes such as helicases. And RNA itself has distinct mechanical properties compared to peptides. So I now want to think about, as we start thinking about this in comparison to SARS, in light of SARS CoV-2. So I already mentioned it has this gigantic 30 kb genome that it brings into the cell. And obviously I think this audience is familiar with this sort of basic simplified life cycle here, where the genome itself must be copied and ultimately prepared for packaging and transcribed into these subgenomic RNAs, which will then be translated into these different structural proteins. And really our focus is in this point of interaction between nucleocapsid and the genome in the early stages of packaging. And our entree into this was essentially noticing that the nucleocapsid protein has all the hallmarks of a protein that would undergo phase separation. It has an RNA binding domain, as well as a highly predicted disordered sequence. And then, of course, there's this magnificently large RNA-based genome. And so we were interested to know whether phase separation could be relevant in the packaging process or in other stages of the viral replication cycle. And I should say our work has been, a preprint has been posted this summer on the bioRxiv, so I encourage you to look into the paper for more details. So if we just begin to look at the nucleocapsid protein, what you can see is that it has, if we look at a estimate, just a computational estimate of disorder in the protein, we can see that there's many regions of the protein that actually are very high in predicted disorder, in addition to having some more folded structure domains that are presumably are known to be RNA binding domains. And if we look at the genome, again, it's this incredibly large genome, 30 kb, that must get packaged. And our first experiment here that I'm showing is that when we mix full-length genomic RNA with nucleocapsid under physiological ionic strength, what we can see is that nucleocapsid on its own forms some small assemblies, but when we incubate it with genomic RNA, we see robust phase separation, the formation of these spherical condensates. when we express the nucleocapsid protein in mammalian cells, it also forms spherical liquid-like droplets that can undergo fusion and relaxation, indicating that they have the hallmarks of biomolecular condensates. We can also see that these form in a concentration-dependent manner. So the questions we've been really focusing on is what features of the genome drive N-protein condensation? The question of how does the virus package one copy of its genome and exclude other RNAs from the cell in this packaging process? How might phase separation relate and be temperature sensitive and relate potentially to both the spread of the virus from bats to humans and also within humans, how this may be impacted by fever? And then finally, whether phase separation could be a druggable feature of SARS CoV-2 replication. So in order to understand what parts of the genome drive phase separation, we essentially analyzed the genome to look for regions that we thought could be relevant for studying this further. So first we looked computationally for areas of the genome that had low synonymous substitution rates, indicating there might be sequences in the RNA that are relevant and under evolutionary constraint among other coronaviruses. We also focused on a region that's been studied in other viruses of the frame shifting element region, the SARS equivalent packaging signal, as well as the RNA that encodes the nucleocapsid itself. So here, the ORF encoding N-protein. And so we did the following, where we mixed fragments of the genome with the N-protein. And what we were really interested and surprised to see is that there was sequence specificity in which regions of the genome could drive phase separation. So the 5' end RNA here promoted robust phase separation, as did the nucleocapsid encoding sequence here, the 3' end, also promoted phase separation. Whereas these sequences in the middle of the genome, the frame shifting region and the SARS equivalent packaging signal, actually led to sort of dissolution of the protein-only condensates that we're seeing. So we can see different RNA sequences have dramatically different effects on the ability of nucleocapsid to phase separate. So we were interested to know how these two may act together, these two different sequences may act together since they're present together in the genome. So we did the following experiment where we mixed together either a mixture of 5' end or frame shifting RNA in a 1-to-1 ratio. And so here you can just see the controls where, again, the 5' end drives phase separation. Frame shifting actually eliminates phase separation. And when we mix them 1-to-1, we actually get sort of an intermediate sized droplet. They're much smaller, and this is indicative of different material states, actually. And so we're interested to know what is actually different between these RNA sequences that either drive or oppose phase separation. And ultimately, what's the function of achieving this intermediate physical state of having these sort of opposing forces present within the same genome? So to approach this, we teamed up with collaborators at UNC, Kevin Weeks and Mark Bernanke, and did a technique called shape map, which enabled us to actually map where the nucleocapsid is binding to the RNA genome. And so in this scheme here, you're looking at a predicted secondary structure that came out of the shape analysis, and in purple are the sites where nucleocapsid protein is interacting, and outlined in black are actually the strongest sites. So this is in the 5' end region of the genome, and this is the area that, again, promotes phase separation. And what we see are a few very discrete, strong interaction sites where the N-protein appears to be binding. And this is in contrast to when we look at where nucleocapsid is interacting with that frame shifting element in the middle of the genome, where we can see that in fact here the nucleocapsid is essentially coding this entire sequence rather than binding to discrete spots. And so this gives us a potential hint that the nucleocapsid is interacting very differently with these two different regions of the genome that behave differently in the phase separation assay. So we've done now some computational analysis of the genome, and this is in collaboration with Rachel Silfan at Princeton, where she essentially analyzed various aspects of the genome for sequence properties in the genome, and compared the elements in the 5' end and the frame shifting region with the rest of the genome. And what was really interesting is that at the 5' end, which we know promotes phase separation, for all of these different properties, we see the most similarity with the 3' end of the genome. And again, this is the other part of the genome that drove phase separation. In contrast, the frame shifting region looks like much of the rest of the sort of interior sequence here, which again, similarly, the SARS element, which is over here, the packaging element similar to SARS, also limited phase separation. And so we think that there's areas of the genome that have really fundamentally different properties here. And so the idea is, our current working model is that the nucleocapsid is interacting at very specific sites in the 5' end and in the 3' end, encoding the nucleocapsid, that is promoting phase separation. Whereas in the middle of the genome, that these are potentially non-specific and electrostatic interactions that actually may be promoting a more liquid material state. We think this is probably very important for packaging such a large genome where the RNA would be highly prone to gelation, and that this may be a way to keep the genome soluble. So why have these opposing features in the genome? I just mentioned one possibility for solubility. We're also interested in whether the physical properties of the condensate could be really relevant for specific packaging of RNAs. In particular, this problem that the virus faces, which is to exclude these highly abundant subgenomic RNAs, but include the specific genomic RNA. And so we hypothesized that one reason to have a particular material state is to provide some specificity in which RNAs are included. And so a simple experiment we've done to begin to test this idea is the following. We constructed droplets made of the nucleocapsid protein plus genomic fragments that are not normally translated, and added either specific genome-specific sequences, so RNAs that should be packaged, or RNAs that are subgenomic or not specific, so they're not actually even present ever in the SARS, that would be host RNAs. And so we did the following experiment. So we created these droplets and then added these different RNAs. And so here you can see the starting state. And in the top row, you'll see genome-specific RNA, and in the bottom row, you'll see the subgenomic RNA added. And so we were really surprised to see a very strong effect here. So these RNAs that we're adding are the same length, they just vary in their sequence composition. And what you can see is that the RNA that would normally be packaged is quite well mixed in these droplets, whereas non-specific or subgenomic RNA is actually excluded from the droplets. And you can see this really nicely, that they are essentially not able to mix well, suggesting that there is a role potentially for the condensate in selectivity of which RNAs are being packaged. And so what we're really interested now is trying to understand how the genome architecture and the spatial organization of these different genome features is relevant for promoting LLPS, and how this may contribute potentially to singularity in packaging a single genome. And so in the last couple of minutes, I just want to talk about how we think phase separation may be really relevant to the COVID-19 spread and also pathology in patients. And so phase separation, as I've mentioned, is inherently, or can be inherently, a temperature dependent process, and we hypothesized that there may be a connection between temperature, phase separation, and viral replication. And so obviously temperature can be a modulator of phase separation, and it's appreciated that bats are at least one reservoir of SARS CoV-2. And as part of their life cycle, they have a natural hibernation temperature that drops substantially and then rises again as they wake up and then are able to potentially then distribute the virus. We also know that temperature gradients exist, at least for the air temperature, in the upper airways of humans, and that there's cooler temperatures in the nasal passages as you move down, the air is warmed. And so we did a number of experiments looking at how phase separation of the N protein may actually be temperature dependent. So first we looked at the protein alone, which again undergoes very minimal phase separation, but a little bit without RNA. And we were really struck by the fact that it forms the most robust condensates at 40 degrees. Interestingly, when we incubate it with RNA, with genomic RNA, what we can see is also a heightened phase separation at elevated temperatures, but we actually see the most robust phase separation at 33 degrees, slightly cooler than body temperature, but potentially relevant to the temperature in the very upper airway. And so this is also really interesting for phase separation biology because there's very few examples of proteins, of biological proteins that undergo this lower critical point phase separation, meaning that they phase separate more at high temperature as opposed to low temperature. So this is actually a very interesting fundamental example for phase separation. And then finally, we've done a few experiments to just look for the potential of whether phase separation could be a screenable process to search for antiviral drugs to treat COVID-19. And so this is really just some proof of principle kind of experiments to show that, in fact, in vitro and then also in cells, we have some experiments in our preprint that small molecules, and here we're just showing some examples of things that we know tend to interrupt phase separated assemblies. Hexane diol is an extreme case. Please don't take this therapeutically, but this disrupts these condensates. Lipoic acid is something that's been known to impact stress granules, also can disrupt the condensates, and interestingly, kanamycin also can interact and appears to change the size and the distribution of the condensates. So what we're interested in here is just showing that this could be a very easy phenotype to actually screen for small molecules that may impact nucleocapsid genome interaction processes. So I'll just conclude here with our model, and I should emphasize it's a working model, but the idea here is that the nucleocapsid protein can readily undergo liquid-liquid phase separation with the RNA genome of SARS CoV-2, and we've identified specific elements in the genome that essentially behave in different ways, those that promote phase separation and those that we think actually work to help solubilize the genome. I think there's a real possibility that nucleocapsid is undergoing phase separation with other RNAs and that this is going to be relevant, that there are likely relevant compartments for different stages of replication. And that this formation of a condensate, while we still don't know or understand how stoichiometry is determined, we think that this is relevant likely for singularity in packaging, but also for excluding non-specific RNAs. We've shown that this is temperature sensitive, and we can imagine and have shown with different small molecules that there's a potential to interfere with this process in a lot of ways. You can think about molecules that might dissolve, that might change the kinetics or the critical concentration of the phase separation, or actually change the composition, the relative amount of protein or what proteins and RNAs are present. I also want to highlight work from others in the community on SARS CoV-2 phase separation. There's a number of really nice papers up on the bioRxiv that I'm highlighting here. And I'll just show our preprint here to thank all of the people that contributed to the work. This was a real team effort between my lab, Kevin Weeks' lab, as well as the Barrick and Sheehan labs at UNC, and also Olga Troyanovskaya from Princeton, who supported Rachel in computational work. In my group, the people that really have spearheaded this work is Chrissy Iserman and Christine Rodan, along with some computational work from Ian Syme. And I really want to thank all of the funding agencies that have supported me generously over the years. And thank you for your attention.
All right. Amy, thank you very much for that story. Great. So I'm curious, you know, as a retrovirus person used to thinking about packaging elements, have people tried putting pieces of the 5' or 3' UTR on other RNAs and getting them incorporated into assembling particles as an assay for a packaging element?
I'm sure people are doing that, and I think we have not yet done these experiments, but I think there are people doing these experiments. And I think, this is, I think the SARS-1 packaging element has been tricky and a bit mysterious in terms of necessary and sufficiency being determined. And I suspect it may end up being the same here. And we've sort of embarked on beginning to discuss these sorts of experiments, but we haven't actually done any ourselves.
And I was also curious, I've been wondering and not seen whether people have looked for host RNAs in virion particles. I mean, are they really just the genome, or is there?
Yeah, I've been looking for this too. So if somebody out there has good evidence, I'd love to hear about it. Yeah, I would love to, I have not yet seen definitive analysis of that. So it would be great to know about that.
Yeah, I mean, again, retroviruses have a lot of other, kind of random RNAs in there, and then others, don't. Like flu is pretty clean, so I don't know.
Right. I mean, there's a real, crowding problem here, right? Like to get that genome in, there's just not a lot of room for a whole bunch of other random stuff. When you just do kind of back of the envelope, size of the virion, and I mean, obviously there's probably some very interesting compaction of the genome that's happening as well in the final stages. But I think there's not a lot of room for a bunch of junk.
Right. Yeah, retroviruses have a lot of room for junk. So anyway, there's a question from the audience about other proteins being in the virion also. So they point out, well, okay, if N-protein interacts with, we just heard with CK2, is CK2 in the virion, and are there other host proteins as well as host RNAs in the virion? Maybe.
Yeah, I mean, I think it's entirely possible, and I think it's also, I should emphasize that what we've not included so far in our reconstitution of this is the M-protein, which is also integral to the packaging process. And the other very important element of this is that it's presumably happening on a membrane or within, a nascent membrane-bound compartment. And so there's, I think, a lot of, potential for the specifics of this to get modulated by other proteins, whether they're viral proteins or whether they're host proteins. There's also evidence that stress granule proteins are interacting with N-protein. And, that's also, a potential role for host proteins, either that's through it's hijacking those host proteins or using them actually to help scaffold condensates, I think is another possibility.
So I was curious what the relationship is of the phase separated, bubbles with the talk about the sort of membrane web replication complexes that are set up. Are they separate, or are they, are the phase separation happening within the replication complexes, or?
I mean, I think, I think we don't know definitively, but my best prediction would be yes, that there's phase separation happening potentially even as part of the formation of those webs, and then also within. There's no reason to think, and there's many different condensates that are forming on membrane surfaces. So there's no reason to say these need to be distinct at all. In fact, the membrane is helping potentially synergize this by serving as a platform so that, the components aren't diffusing in 3D, they're diffusing in 2D. And so that essentially lowers the critical concentration for formation. So I think they're very, probably coexisting. The challenge is the scale of these in cells is much smaller than what we're able to see when we reconstitute this. And so assessing the puncta that you see in cells for liquid-like properties, for all the features of a phase-separated assembly is probably impossible, if not incredibly difficult, right? And so I think that's probably one of the biggest challenges, is definitively showing where in the pathway this is the type of assembly is relevant. And it may be at multiple steps, it has different features and different characteristics.
Sure. No, I think that's a common issue in phase separation everywhere, is that whatever you do in vitro is hard to relate to whatever is going on in a cell. And yeah, often, as you say, the scale is different, and the requirements and the setting are very different.
Right, exactly.
So we have one question from Laura Landweber here about whether there's better understanding of the properties of the packaging area, like GC content, or she says Kmer frequencies. Those graphs showed a lot of features of the RNA, and with some that may be sort of mapped with packaging, but I guess she's asking for other issues like GC content and things.
Right. So, I mean, certainly GC content does, and I'd say we're still in the process of really trying to dissect what makes these sequences different. There's really nice work also from Anna Pyle's group at Yale, looking at shape map structure analysis of the whole genome. And there's a lot to parse there, actually, I think. We see that the nucleocapsid, not necessarily at I don't have an answer for Kmers at this point, but what we've been focusing more is on not necessarily the sequence itself, but the structure. And the structure does seem to have sort of a motif of a single stranded area that's flanked by two structured elements. And that seems to be kind of the preferred binding site. We haven't done proper EMSAs to do affinity analyses to really see what the affinity differences are for those structures, but that's something that we're working on right now, as are, I'm sure, many other people as well. So I feel like we're just kind of in the early days of getting to understand like what the features are that are really distinguishing it.
Right. Well, the HIV RNA packaging element's been studied for about 20 years, and we still don't know really exactly what is being recognized. But people like Mike Summers and others are I think zeroing in on the features that make the nucleocapsid protein there bind best. But
okay.
Yeah.
Well, that's comforting that we haven't figured it out in the last couple months.
Time, a little bit of time. Okay, well, that's all I'm seeing there. I guess we'll just be listening for the next steps. And
okay, yeah, stay tuned.
Thank you for a beautiful talk.
Yeah, well, thank you so much for the opportunity. I really appreciate it.